6th in our Broad at 15 talk series. The Broad Institute came into being in 2004. Since that time, we've witnessed some amazing progress in many fields of biology and medicine. And these talks are basically a little way of celebrating that amazing pace of scientific discovery and the parts that the Broad Institute have played in it. It's my honor to introduce our speakers for this evening, Deb Hung and Pardis Sabeti. Deb is a core institute member of the Broad, co-director of our Infectious Disease and Microbiome program, a professor of genetics and of medicine at Harvard Medical School, and an infectious disease specialist at Massachusetts General Hospital. Pardis is an institute member and a professor of organismic and evolutionary biology at Harvard University, as well as of immunology and infectious diseases at the Harvard T.H. Chan School of Public Health. Together, they're going to talk about how scientists are using advanced genomic and chemical tools and working with both individual patients and global communities to tackle the drug resistance and infectious diseases. If you have any questions for Deb or Pardis after their talk, please raise your hand for one of our mic runners or text your question to the phone number that'll be on the screen. And if you're watching online, please submit your question by YouTube chat. And if you happen to be tweeting tonight, we just ask that you please use the hashtag BroadAt15. Without further ado, Deb Hung.
So thank you so much, Tom, and thank you all of you for coming to this Broad at 15 talk. Pardis and I greatly appreciate your willingness to come and hear about a topic that she and I are extremely passionate about. So as you all know, infectious diseases is as old as time. Not only have historical records shown evidence of plagues that have wiped out populations throughout history, but there's actually been evidence of different pathogens that have been discovered in different historical places, including mummies. And they've found evidence of different diseases, including tuberculosis, smallpox, leprosy, and the flu, and many others. There's also been a historical record of many very significant events that have been instigated by infection. Many of these you know. So for example, the Black Plague in Europe, where the reports is that it knocked out 30 to 90% of the population in Europe at the time. There's also the episode of the introduction of smallpox into Central and South America, to which has been attributed actually the decimation and the elimination of the Aztec Empire. And then, of course, more recently, many of us have heard about the Spanish flu outbreak in 1918, which, again, the estimates are that there were about 50 million deaths. Fortunately, through this time, we've actually made some progress in gaining insight into infection. And probably one of the first people that some insight was attributed was actually Fra Castaro. In the 1500s, he was a philosopher and scientist who was the first one to suggest that there are actually diseases that can be passed from individual to individual. But it wasn't really until the 1880s when Antoine van Leeuwenhoek invented the microscope that one could even begin to comprehend or begin to get some view of what microbes are. And then it wasn't really until the 1860s when Louis Pasteur and Robert Koch came up with the germ theory, which then really changed our understanding of infection altogether. And the two of them, and really Robert Koch, came up with these rules to suggest that to establish the evidence of an infectious pathogen and an agent that can cause infection, you have to be able to isolate it from a patient, reinfect that agent into someone else, and basically recapitulate those symptoms and then re-isolate the organism. And after that, Robert Koch then went on to actually identify many of the major pathogens, including bugs like tuberculosis and cholera. So really, in the following decades, there was the discovery of numerous pathogens that cause disease in humans. At the same time, scientists were making progress in terms of how to treat infection and actually to decrease mortality from it. While the concept of vaccines existed before Edward Jenner did his seminal experiment with cowpox, he's really the one who's attributed with this idea of being able to take an agent from one person, introduce it into another, albeit at low levels, and actually induce immunity so that the immunized person doesn't get disease. So while people had been doing this and really scraping lesions from people with smallpox and introducing it into other people and showing you could get protection, his seminal experiment was he recognized that women who were taking care of cows that had cowpox, actually were now immune to smallpox. And so he actually went on to show that he could scrape lesions from cows, introduce it to humans, and protect them from smallpox. That's vaccines, but at the same time, there was progress made in the ideas of developing small molecules or drugs that could similarly combat infection. So Paul Ehrlich is the one who is attributed to this concept of the magic bullet. The idea that you could have a molecule that could be specific and target something over something else that you didn't want to hurt. And the idea that you could have a magic bullet that would target the infectious pathogen and leave the human intact was really his idea that has now been played out dramatically, right, in the concept of antibiotics. Again, in 1928, you probably all have heard this story now of Alexander Fleming and this idea that he had left a petri dish in his lab open and that there was a mold growing on it. And when he came back, he found that, in fact, that around the mold, bacteria couldn't grow, and that was the first identification of penicillin. In fact, it was not Alexander Fleming who went on to discover the actual structure of penicillin, nor was it he who went on to discover how to produce it in mass production so that you could actually use it on patients. But that was really, though, the beginning of what was then called the golden era of antibiotic discovery. So you can see in the next subsequent few decades that there are many different new antibiotics with different mechanisms that were discovered. And it was so successful that many people declared victory. So here is a quote from the Nobel laureate, Sir Macfarlane Burnet in 1962. Almost all of the major practical problems of dealing with infectious diseases has been solved. And a US Surgeon General in 1967 said the war against infectious diseases has been won. Now, I hear a few chuckles, perhaps because you read the headlines this morning to see what the latest count was on the coronavirus. But how wrong they were and how premature those declarations were. Because, in fact, we know that infectious diseases is still one of the leading killers in the world. Globally, you can see that infectious diseases still is actually the fourth leading killer. Whereas in low-income countries, in fact, it's the leading killer. But why have we made so little progress in infection? Right? Why is it so hard? And I will argue that there's several factors that have made it really challenging to try to more or less win the war on infectious agents. The first is that we continually have new emerging pathogens that we had never known about before, or ones that were and we thought we were done with it, and yet now they're newly emerging. So to give you a sense of the scale of that, in the past 30 or 40 years, there have been probably 40 or 50 new infectious agents to have been identified and reported. The second problem, though, is actually is what I've described. Infectious agents, as you know, are transmissible. They can be spread. And so not only can these agents appear in one place in the world, but they can spread easily globally. So just to give you a few examples of that, in 2010, you might have heard about an episode where UN workers actually traveled from Nepal, where they were working, and moved to Haiti, where there was incredible need. And they brought with them cholera, and actually introduced cholera into Haiti and set off an epidemic there. Similarly, you've probably heard that HIV probably originated in Africa, and yet, as you know, it's global. There is not a single country that's been spared. Finally, a third example about an antibiotic-resistant organism. So there is an organism called Acinetobacter baumannii, which we have known about, but notably, that there were soldiers returning from the Gulf War who actually brought back a resistant form of that particular bacterium, actually because it was endemic in the Middle East, but not yet in the United States. The other thing that makes managing and trying to tackle infectious diseases so challenging is that there is enormous heterogeneity in agents that can cause infection. They will range from, some of these pathogens you can see up here, West Nile, Lassa fever, Ebola, HIV. These are viruses. And what viruses are is they are infectious particles that can be spread from person to person. But they actually can't survive on their own. They actually have to infect another human cell or another cell type other than themselves in order to replicate and then to cause disease. And it can range from that to the bacteria that I suggested to you, cholera, Acinetobacter baumannii, TB. These are all bacteria which are unicellular single cells which can survive free living on their own and yet, as you know, can cause terrible disease. And I'm not even including other parasites like here, Cryptosporidia, malaria, right? So you can imagine that from a genetic standpoint, it's extremely diverse, the types of agents and organisms that can cause infection. So the idea of trying to find one drug that can rule them all is impossible. Just to give you a small sense of how different these are, 2 different bacteria, so I'm not even talking viruses or parasites, but simply 2 different bacterial species are far more different than we are from corn. Okay, so let me describe. So not only are there new agents, but here's another problem. And I will argue that in some ways infectious diseases is the one medical discipline that we're moving backwards. So let me illustrate that by describing 2 different patients. The first is a 33-year-old woman with blood poisoning from streptococcal infection, meaning there was bacteria in her blood after a miscarriage. She was given antibiotics and she survived. The second patient is a 35-year-old woman with a bacterial pneumonia due to this bug called Pseudomonas aeruginosa. She was told nothing could be done, and she died. The first patient was in 1942. That was Mary Miller. She was the first woman who was treated with penicillin, and she actually only died more recently at the age of 90. The second patient is someone I saw last year in the hospital. And I was the one who had to tell her that we have no antibiotics left because the bug she has is resistant to everything we have. And so I had shown you this timeline, right, which shows this golden era of antibiotics discovery. But here's the problem. On the bottom of the timeline shows you when clinically resistant antibiotic resistance has appeared. And so there is no antibiotic that appears above where its matched color doesn't appear below. The bugs are too smart. They can evolve too easily and become resistant to the antibiotics that we have. And so what is the consequence of this? Well, the UK actually commissioned a report to be put together by an economist of all things, Lord Jim ONeill. And he was trying to predict if we continue on this current trajectory of increasing antibiotic resistance with an inability to discover new antibiotics, he projected by 2050 there would be 10 million deaths per year from antibiotic-resistant infection. Currently, it's hard to know because we don't actually keep hard, fast numbers, but the estimates are worldwide. It's probably on the order of 700 to 750,000. But the projection is we would be hitting 10 million. It would become the leading cause of death in the world again. And economically, the cost would be $100 trillion. Just to give you a little bit of a feel for how easy it is for bugs to become resistant to infection, I'm going to show you a short video that was put together by a friend and colleague, Roy Kashani. He was at Harvard Medical School when he made this. He's now at the Technion in Israel. But what this I'm going to show you is basically this is a big plate. And at each of these divisions, on the outer edges, there's no antibiotic in the plate. In the middle is the highest concentration of antibiotic. And so you can see that there's basically gradations as you go across. And so what you're going to see is Roy seeded the edges with a sensitive strain of bacteria. In other words, the antibiotic should be able to kill it. But in a matter of days, you're going to see what the bug can do. So the white is the bugs, and this is basically that you can see that they grow in right easily where there's no antibiotic. Oh, now you see, right? A bug or few bugs have become resistant. They can grow in at the next concentration. At the next concentration, some of them spread. And you can see pretty soon they can grow all the way on the highest level of antibiotic that's in the plate. This is a matter of days. When you went from something where the antibiotic worked to the antibiotic. The bug is completely impervious to the presence of the antibiotic. That is what the evolution of antibiotic resistance is like. So, have I depressed all of you enough? Genomics, right? You have now all heard about the revolution. What can it do? Can it change things at all? So probably most of the attention about genomics is focused on the sequencing of the human genome, and that's where all the attention came out, that the first map of it came out in 2003. In fact, in 1995, the first full genome came out for a single bacterium. And you can see then that since that time, we've had increasing numbers of genomes that have become available for different bacterial species. Here it is, now we're topping 14,000 by 2014. And honestly, what happens after 2014, I'm not even sure anybody keeps track anymore. And the reason they don't keep track is because it's just too easy to sequence, so everybody can do it in their lab these days. To give you a sense, kind of back of the envelope, on a single instrument, a single sequencing instrument, you can get about 10,000 bacterial genomes in a day at a sequencing cost of about $2. So you can see everybody is doing it now. How does this impact infection? So let me give you a little bit of a timeline of pathogen discovery and give you an example of how the introduction of genomics and the incredible power of sequencing and the development of other kind of parallel technologies at the same time have, I think, changed how we can approach infectious diseases. So on the top is tuberculosis. Right? I told you this is the bug that's been found in mummies. Just for the record, it still is now the leading cause of death worldwide from a single infectious agent. But here you can see that people were infected way back, right, millennia ago, and yet Robert Koch only discovered the bacterium that causes the disease in 1882. And then it took several years after that where actually they developed a diagnostic so they could actually test people and figure out who had it. All right, now fast forward to HIV. The first report of it was in 1981. A few years later, Robert Gallo actually then reported the virus, the HIV virus, which causes it. And another year to develop a diagnostic test. So now we're on the order of years. Okay, now let's move to the beginning of the genomic revolution, SARS. Probably most of you remember that. So the first report of it, of folks with unusual pneumonia, was in November of 2002. The discovery of the agent in March of the next year, months. And the first diagnostic and sequence and test came out in April. And I remember at the time when this was happening, I was stunned. I'm like, wow, good job. We should pat ourselves on the back. But let's fast forward even more to something I bet you guys are all following quite closely now, right? And that is the COVID-19 virus, this new coronavirus that's been reported. So the first report, December 31st of last year. By January 5th, the report of the discovery of the agent and the first sequence was made. By January 13th, the reports of the first diagnostic test. Now we're in the realm of days for discovering a new pathogen, having its sequence, and be able to turn its sequence into a diagnostic test. A little bit game-changing. What about diagnostics? As I said, right, if we have the sequence, we can now turn it into a diagnostic test. The standard in microbiology labs these days is not yet completely leveraging all these genomics, but that's where we're headed. Many of the approaches are still based on old lab methods. Quite frankly, many of the methods that Robert Koch used in the 1800s to discover pathogens. If you have a fever and go to a hospital and they draw your blood and urine and these kinds of things, they will still be doing this, culturing. What's the problem with that? The problem is that we often get an answer in days. And that doesn't seem so bad, right, compared to before we couldn't even diagnose. The problem is that there are some infections. There is a clinical syndrome called septic shock, which is triggered by an infection. And every hour that you don't get the right antibiotic, your mortality increases by 7%. So you can imagine if you don't get an answer for 1 or 2 days, imagine what the mortality is like. There's also other methods to diagnose, and these are kind of the methods that people have been using for a few decades. And that is to draw blood from a human and look for human responses, antibodies. The problem is that test can take weeks. Where are we now? What is the genomic revolution allowing us to do? Well, so there has certainly been an interest in, rather than waiting to isolate the bug and wait for it to grow, rather than measuring human responses and these kinds of things, it's to say, can we detect other pieces of a pathogen where we don't have to grow it in lab? People have developed tests to go after proteins, sugars, other components of pathogens. But what's the really attractive one is actually the nucleic acids, because that is more or less a map that distinctly identifies a pathogen. And so many tests have now become available because of the discovery and development by Kary Mullis of something you've probably all heard of, is polymerase chain reaction, PCR, for which he got the Nobel Prize in 1993. But the idea is that we could now amplify sequences of DNA that uniquely identify that pathogen. Now, it's not always trivial to design the fragment that we go after, because it has to be completely unique to that bug. It can't cross-react with sequences from other, from the human, much less other bugs, because you want it to be sensitive but also specific. But nevertheless, that is being implemented now and is transforming to get a more rapid answer. But people are now beginning to think, well, let's even leapfrog over that. Sequencing is so easy now. It can be cheap and continue. The hope is the sequencing costs will continue to be cheaper. And so people are saying, you know what, why don't we just take the sample that we get from the human and just try to sequence it directly? Now, the computational side becomes a little bit of looking for a needle in a haystack. Because, for example, if you draw a sample from blood, the signatures, the sequences from the bug may be 1 out of 10 to the 9th, which come from the host. And so it's looking for that needle in the haystack, but both the technologies to sequence that deeply as well as the computational methods to find that needle are improving, and people are now beginning to implement that in the clinics as well. Okay, that's about identifying the pathogen. What about treating it? And that's a little bit of a different ballgame, because it's not enough to know what bacterium, what species you're infected with, right? We actually want to know what antibiotic can we use. And as I showed you in the movie, the bugs can evolve to be resistant to many different antibiotics. So we need to know which one works. This gives you an example of what it looks like in the lab on a plate. So in each of these small white circles is impregnated a certain antibiotic. The white, again, are the bacteria, and you can see around each antibiotic disc, there's clearing because the antibiotic works on those bugs. Here you can see that around these discs, the bug can now grow in right next to the disc. It means these guys are resistant. How does the bug go from this, where it works, to resistant? It can be as simple as picking up a single mutation. Right? You guys watched it in real in the movie, right? But it can be that simple. Pick up a single mutation and you can become resistant to an antibiotic, or you can pick up a resistance gene, because bacteria can often kind of swap their genes in a neighborly fashion. So this was the way, this has been the classical way of whether we know whether an antibiotic works or whether it doesn't. And so, again, what the genomic revolution is allowing is to say, you know what? Rather than waiting for it to grow, I'm just going to actually go and query, does it have the mutation or does it not? Can I use that to quickly tell me if a bug is sensitive or resistant, whether the antibiotic works or doesn't? And in some cases, that works very well, and there are actually a number of tests out there that are being implemented now, where they're simply looking for the gene or the mutation that confers resistance. But here's a problem. The problem is that this works if you have a comprehensive database of all the mutations and all the genes that can confer resistance. And while that, as I told you, it works in a few cases, such as tuberculosis, in the mass majority of bugs that cause human infection, we actually don't have that comprehensive database. And so, we need other methods. And so there are many other labs, ours included, here at the Broad and worldwide, who are trying to find alternative solutions, right, where we can in fact figure out if bugs are sensitive or resistant much faster. Okay. Now I'm going to project a little bit, to the future, because now I'm going to talk to you about we're really at the edge of what I hope will be the next kind of revolution in diagnosing infection. Because everything I've told you thus far is actually testing the pathogen itself, the bug, right? Whether does it grow or not on an antibiotic, does it have this mutation or not, right? It's all about the bug. Whereas there's a whole new push to say, sometimes it's really hard to find the bug. Right, you guys have probably had this frustration of you're sick, you have fever, you know you have an infection, you go to the doctor and they're like, okay, we don't know what it's coming from. The idea is, okay, we can't find the bug in the tests that we perform in the blood that we draw, but maybe your own body, right, the human body can be a sensor to help diagnose. So there is kind of this whole push now to say, can we actually draw blood, can we measure host response genomically and actually ask the host to read out to us what is the infecting pathogen? And I think we're on the beginning edge, and I'm hopeful in the next years to decades that actually is going to become reality. So that's the impact of genomics and novel technologies on diagnostics. So let me just come to the last kind of topic that I think there is the potential to transform. But I will admit that it's a bit behind diagnostics and pathogen identification. So that is, what about finding new drugs? Is genomics and the ability to sequence and the new technologies associated with it, is that going to change our ability to find new antibiotics? So as many of you know, a common analogy for the genome, and a genome of a bacterium or a human, is that imagine the whole genome is a book. And that genes are sentences within the book, and that the genes or the sentences make sense because they're encoding a protein or some important functional unit in the cell. And then the sentences are made up of individual letters, which are the nucleotides. So what is the genomic revolution allowing us to do? It's actually allowing us to define what are all the genes, right? What are all the possible sentences, right, within a pathogen? But even more than that, what we can currently do is we can actually go in and systematically knock out every one of these genes. So for example, in a typical bacterium, there might be 4,000 genes. So we can go in and systematically knock out every one and say, oh, if we knock this out, can the bug live or can it die? If it dies, it shows us that that's an essential gene, and it means that it's a vulnerability of the bacterium. It's an Achilles heel, which we might then develop drugs that target that. The analogy is to an automobile, where you can define all the parts of the car, but they're not all equally essential, right? You don't really need those leather bucket seats, but you need the spark plug. Right? And it's the same idea. Can we figure out what are the essential parts that we could potentially develop antibiotics against? So kind of after the first bacterial genome was published and people realized they could do this, there are a couple companies that jumped right on this, thinking, wow, this is really going to transform antibiotic discovery. Unfortunately, it hasn't been so easy. So in 2007, GSK, GlaxoSmithKline, reported their 7-year experience of antibiotic discovery in the post-genomic era. They invested $200 million and came up with few good candidates for new antibiotics. Similarly, AstraZeneca reported their experience over 10 years. They weren't so transparent about their investment, but again, very few candidates. Disappointing, yes. Is that the end of the story? No, I don't think so. Because what's important to know is that these guys performed these genomic experiments very early on after the first genome was available. And so a lot of their analysis of what are the essential parts in a bacterium were done based on a single genome, a single strain, as opposed to understanding the heterogeneity and diversity of different species. It's like saying, that you are identical to the person sitting next to you. Clearly, that's not true. And so by trying to find targets that are vulnerable based on a single genome is probably where they failed. And so now, as I suggested, when we can get 10,000 genomes of the same species in a single day, you can imagine that it's time for us to revisit that. And so again, here at the Broad and many other places, we're kind of revisiting that idea with the belief that at this stage, because genomic technologies are in a much more advanced state, that we will in fact be able to hopefully change the antibiotic discovery process. But this is to say that it's not, we admit it's not easy. And why isn't it easy? It's because bugs are smart. They have evolved to survive out in the wild and to protect themselves. And so they have very unique cell walls, meaning the outer part that surrounds the cell is really hard to get small molecules into. They don't want to take up foreign molecules that have the potential to kill them. So this is an example, right, of a human cell. There's a single membrane, and so drugs can get in. A gram-positive, one type of bacteria. If you've heard of MRSA in the news, that's what this is. But you can see it has these extra layers. Gram-negatives, such as the Pseudomonas aeruginosa that infected the woman that I told you about, that I had to tell last year that we didn't have any antibiotics for, not only has extra layers, but it has efflux pumps. So if molecules do make it through all these layers, it has a system to pump them out. And so it's really hard to accumulate these, these small molecules and drugs inside the cell where many of them need to work. Just to give you an example of what that means, if you had high blood pressure, if you had high cholesterol, and you get started on a drug that's going to be targeting your human cells, many cases you're started on a drug for about 10 milligrams. There's an antibiotic that we use to kill bacteria. When we give it to the patient, we give them 24 grams, 5,000 times more. And you can imagine if you have to give people such high doses, you can imagine that the side effects will add up. And so that's actually what complicates and dooms a lot of antibiotic discovery. So when you take all these factors together, make no mistake, discovering new antibiotics is hard, but you can see the hope is with introducing new technologies, which we have at hand, that we're going to be able to change this process. This is just to give you an example, right, that antibiotic discovery against any of the pathogens that I've talked about today is hard. And it's hard not simply from the science, but it's actually hard from the economic standpoint, from the regulatory standpoint. It is as close to what I would use as an analogy as trying to take on climate change. It's a political problem. It's a global problem. And so, as someone said to me recently, asked me, do I think that the problem of antibiotic resistance can be solved? My answer is, do you think that climate change can be solved? And their answer was, it needs to be solved. And that's my response. The problem of antibiotic resistance needs to be solved, but it's extremely complicated. And this is just give you an example of the economics. So this is a timeline of what it's like for a company to try to invest in coming up with a new antibiotic. The first number of years, right, they're just sinking money in doing research to discover it. Then there's the clinical research. They're still going in the hole. Okay, now it's on patent. They're beginning to sell it, barely trying to climb out of that well. And at best, when they break even, the patent is gone. And in fact, the last few new antibiotics that have been developed in the last few years, there's one case that within weeks of their getting an FDA approval, and then another case where they didn't, they were shutting their doors within days. So this is all to say that it's an enormous, complex problem that needs to be tackled from many different aspects, the science being one of them. And then finally, I'll leave you with, again, the future of where I hope we're going with all these technologies to change infectious diseases. So again, everything that I've talked to you about, antibiotics and getting into the bugs, is bug-centric. It's focused on the bug. But let's not forget that really what infection is about the human patient dying. And so if that's the case, how come we're focusing only on the bug? It's the only field, as I said, where we're losing our antibiotics because of resistance, and yet we are not looking for any new ones that would actually improve mortality. So if we remember that that infection is really actually a complex dance between a host and the bug. You can imagine looking for ways to intervene actually on the host side that will tip the balance towards the host and not simply just focusing on the bacteria. And indeed, if we found things that targeted the host, because evolution is much smaller, the hope is that resistance would not be such a big problem. Okay. So let me end by saying that what I focus on is infectious diseases as it's impacting the patient. It's the clinical medicine side. But let's not forget, because it's transmissible, it has a tremendous public health aspect to it, and that is what happens when you think about infection on the population scale. And so I'm going to turn it over to Pardis, who's going to give you some case examples, actually out of the Broad, but we'll hopefully give you a concrete idea of how genomic technologies are actually changing management of infection on the population level.
Thank you all. It's really exciting to be here and to get to do this with Deb, one of my favorite people in the world. It's an amazing experience to be here at the Broad, an institute that's only been around for 15 years, that is built on all of the science that Deb described for a long time. And we are in a place where we're really kids in a candy store, where for decades people have been making these observations of the natural world, painstakingly trying to identify new phenomenon. And here we are in a place where we actually can truly transform and potentially, and of course I always know that the infectious diseases, the viruses and the bacteria and fungus and other elements are so tricky that I will never say we can definitively stop them, but I think that we can live in a world in which we can make a major impact, we can change our relationship. But as Deb said, I'm going to give you an example from the Broad of where genomics can become so important and where the need for it is so vital. And this is from the Ebola outbreak of 2014. And this is one of the great outbreaks in recent history that had left a real mark on the population. We're sitting right now in another one, in another, for a many terrifying time, thinking about what could happen if an infectious disease spread through the population. In March of 2014, the world became aware of Ebola in West Africa when about 60 individuals were identified. Patient samples were sent to out of West Africa, out of Guinea, where the outbreak started, to Europe, where they were detected to have Ebola. But the fact of the matter is that outbreak didn't start then. It didn't start in March of 2014 when epidemiologists went back and tried to trace when it started. It's believed to be, in our presumed index case, is a little boy named ONeill in December of 2013, about 4 months before the outbreak was declared, came down with a mysterious illness after playing in a tree inhabited by bats. And soon ONeill became very ill and passed away. And then his sister became ill, and then his family and his loved ones in the village. And by that time the outbreak was detected 4 months later, it had already spread through districts throughout Guinea. By April, it had reached into Liberia and really spread across the region. And then by May, it came into Sierra Leone, but at that point, it came in like a tidal wave. It spread throughout the entire region. And if you look at this from sort of the number of cases that emerged, early in that outbreak, we were already seeing one of the largest outbreaks of Ebola in history. And there are many, many cases coming in. But in May, the trajectory changed. There was an inflection point, and suddenly you saw this escalation. And a lot of people suspected that this had to do with the fact that Sierra Leone had regions that were even more populated that it was getting into, that MSF, WHO, and other organizations had already tapped out the resources they had dedicated, and there really wasn't anybody coming into Sierra Leone to stop it. But it definitely felt like a rapid progression. And my team was well aware of this, and this is how the Broad found itself at the epicenter of what was going on, because we had been working in Sierra Leone for many, many years before, for about 7 years before this outbreak started. We'd helped the hospital establish diagnostics for another hemorrhagic virus called Lassa virus that spreads in that region, in Liberia, in Guinea. And in fact, the Kenema Government Hospital where we worked was a center for excellence for that entire region. It was doing diagnosis of Lassa for all 3 countries. So we knew that if Ebola was coming to Sierra Leone, that we would see cases. And so we rushed out. Members of my team on the right, Christian Anderson and Stephen Geyer, met Dr. Humar Khan and Augustine Goba and the team there Dr. Khan is the head clinical physician at that hospital, seen more cases of Lassa or hemorrhagic viruses than anybody in the world. And we helped them set up and establish diagnostics using PCR that Deb described. We also had, for a long time, set up the ability to take the discarded clinical samples that we collected, deactivate the virus, and send them to the Broad where we would do sequencing. And so we use that same premise, working with the Sierra Leone government, to send samples to the Broad and to do analysis. And what we saw within 99 patient samples that we got within a matter of weeks when this outbreak hit, we saw hundreds of mutations that popped up just in a matter of weeks. And that is really important because fundamentally, as Deb also described, these inform diagnostics, they all inform vaccines, they inform therapies. All of those are fundamentally based on the genome sequence of any organism, in this case, the Ebola virus. Is using that genome sequence to tell what kind of antibodies get created, what kind of proteins get created that are targeted by antibodies. And so that information is really critical to being able to move with the target as it moves. It also allows us to start looking at other things. We can analyze that genome sequence from these 99 different patient samples, and in comparison to 3 genomes that were published from Guinea in March. And what we saw, looking at essentially this, what we call a phylogenetic tree, it's the family tree of these viruses, is we saw that all of them seem to be very closely related, so closely related that it appeared to be a single transmission into the human population, and from there was transmitting from human to human. And that was a very, the two kind of simple things that we got from this data are things that very simple. One, the virus is mutating, something we already know, but the data really shows, hey, this is a moving target. And 2, that it was transmitting from human to human. And that was very impactful because early in the outbreak, most of the public health messaging that we saw were with a feel that it was coming from the environment. So don't touch monkeys, don't touch bats, don't touch fruit that have been touched by these animals, don't eat a lot of the foods that you're also eating. And so that was really where the public health messaging was going. But fundamentally, after realizing, oh, this is just a human-to-human event, we started seeing more messaging around hand washing, not touching, contact tracing became more and more important. And so we can see this fundamental, simple thing that we can share. It also allows us to take a close look at those mutations and say, what are those mutations? And the vast majority of mutations that happen in any genome don't really matter. They're nature testing things out, or they're just cleared from the population. But every once in a while, something interesting may occur. And what we saw is in the protein, the glycoprotein of Ebola virus, it's coating the surface of the virus. It allows it to bind to human cells and enter. We see an alanine to valine change, a single mutational event. And that, as it turns out, we partnered with Jeremy Lubin at UMass Worcester and partners in the UK, Jonathan Ball's group, and here at the Broad, we were able to show that that single mutation in culture increases the infectivity of the virus. It actually increased its ability to bind to human cells and enter, as well as in other non-human primates. And that's consistent with something we see about the receptor in that particular, in non-human primates and humans. But it actually decreased its ability to bind to other mammalian cells. And so the data, and it's still in development, but what it's suggesting is that as Ebola had more opportunities to transmit from human to human than ever before, it was increasing its ability to do so. And that may be one of the many reasons that this outbreak went on to be the largest outbreak we've ever recorded in history, more than 30,000 confirmed cases, more than 11,000 confirmed deaths. Just mass devastation in the region, and a real stark wake-up call to the worldwide community of how precarious this all is. And this is particularly impactful for us and for others because of the great toll of healthcare workers. They're the ones at greatest risks. They're the ones that are exposing themselves every time that they connect with a patient, or families that are caring for loved ones. We saw Dr. Khan pass away during this outbreak. We saw many other healthcare workers at Kenema and around West Africa perish in a region that already has too few of them. And so it's a great tragedy. But then we also think about the fact that it was a tragedy and it could have been a cataclysm. This is Christian Happi, a longtime Broad Institute affiliate and community member. And this is us sitting about 100 yards away from Christian's lab at the time at Redeemers University, which is on the grounds of Redemption Camp and on the same grounds of Redemption Church, about 100 yards away. And just days after Dr. Khan passed away and several other nurses became ill, Christian called me to tell me that he diagnosed the first case of Ebola in Lagos, a city of 21 million. But to make matters worse, he was rushed off the call because he had to get ready for First Friday that was coming in, the annual event that would bring millions to the steps where we sat. And so we think about that, just that close call that occurred and what would have happened if a single case of Ebola had moved through that kind of a crowd. That is what always reminds us of how dangerous this whole situation is and could be. And so when we think about it, we think about a few principles of what we can do that genomics can bring to bear. It's about detecting, detecting cases of infectious disease anywhere in the world as soon as they occur, about connecting that information across a grid, sharing that information in real time so that we can start identifying clusters of infections that may be related, about engaging everyone to come to attention, to kind of incentivize people to come and seek care and be accounted, and to empower every actor in that system so that they don't feel helpless, that they know what to do, where to do at the right time. And ultimately, all of this is to work together to buy time to come up with a way to overcome. When we think about detection, this is our team at the Broad Institute that was able to do that sequencing rapidly. We were able to sequence the virus within just a matter of days, just a couple of weeks, and start sharing that data to the cloud. And we use that same process, the genome sequencing, that allows us to capture everything in the sample that Deb explained. But that was a large effort. We're now moving as the technology gets better and better. We also want to move it so that the sequencing happens in country. A lot of the time, we wanted to make a table of how long it took for the sequencing to happen. Most of it took just getting it out of the country. Well, we need to be able to do things on ground. And so we've worked with Mass Design firm to establish this is about 80% done, but it's a genome center that's designed to work in a rural part of West Africa, but to have all the technologies that the Broad Institute comes to bear that are critical, and partnering with the Broad to develop that. And that's really important because of this thing that Deb described, this ability to just take a sample and sequence everything in there. And so in those cases, you don't have to know what you're looking for. But it is expensive, relatively, and it isn't something everyone can do right now. But the fact of the matter is we didn't need that in the case of Ebola. We knew that Ebola was circulating in Africa since 1976. And there's ways that we could have known that it was already circulating in West Africa, or that it might be. Also, in the case of Zika, it became a major outbreak just after Ebola was sort of under control. We saw Zika emerge, but we actually knew about Zika since 1947. So for the vast majority of viruses that we actually are concerned about, we've seen them before, could have seen them before, not just in the environment, but in clinics. And so how could we do better about building diagnostics to test for things anywhere in the world? Well, when we have those genomes even sequenced, we can use PCR, as Deb was explaining. This is a picture of the first gel from the Ebola outbreak. We got this email from Augustine Gobe, pictured here, saying, hey, is this what I think it is? And that was the first confirmed case in Sierra Leone. And that was the diagnostics that the team helped establish. Fundamentally, if you know the targets you're going for, you can design assays, you can amplify that part of the genome to say, here, this is what we're looking at. But now, we need even more sensitive diagnostics, ones that can be developed within days but can be done anywhere in the world. Because we see this, in Zika, just the fear people have of not being able to get what they need where they need it. So we need field deployable, inexpensive, fast, programmable diagnostics. And this is a place where the modern revolution in genomics and molecular biology has become to bear. One of the biggest technologies of the last decade is CRISPR. It's transformed a lot of how we think about molecular medicine and our ability to affect change in human health. But fundamentally, CRISPR is identified as a bacteria's immune system to viruses. It allows them bacteria to circulate guides around them, and see if a particular guide targets a virus, it will detect and destroy it. And so fundamentally, CRISPR was designed to detect viruses and to then cut them and destroy them. It pulls in a protein, and at that point, a cut occurs. But there's a particular type of CRISPR enzyme, Cas13, that can cut at a particular target and an RNA target. And then when it does that, it actually starts cutting everything around it. And if you actually bind that to a fluorescent probe that's tied to a quencher, if it identifies the thing it's looking for, it starts cutting wildly, it can read out immediately. And this can be done so sensitively and in such a simple way that it could be read out on paper. And so this allows us to get to the point where we could have diagnostics anywhere. The next thing we want to do is connect. During the Ebola outbreak, information was shared on paper, by Excel, by email, if at all, and it made a coordinated response really difficult. But if we actually have national genome centers or regional genome centers and hospitals that can do larger diagnostics and village clinics where we can have paper-based things, we can actually try to find ways of connecting that information on a grid. And ultimately, what we want to do is be able to have everyone upload information on a cloud in real time. And we're beginning to do this in Nigeria, at sites in Nigeria. We're seeing, and throughout with the Africa CDC and the Nigerian CDC, partnering to essentially have these types of diagnostics everywhere and start sharing that information on a cloud-based system. The Broad has, in this kind of after Ebola, worked to establish genome centers in Senegal, Sierra Leone, and Nigeria, and to help advance one in Liberia, and all sharing this kind of information. The next thing we want to do is engage. So let's say we have that system, but what if people don't come to attention? And one of the places where we thought about how important and how rapidly we need people to come to attention happened closer to home. So I'm part of Harvard University's Outbreak Response Committee. If an outbreak is declared either at the university or around, a group of us call to attention. And we suddenly got called to attention when in the sports center we saw a number of sports teams that were coming down with mumps. Then we saw it moving into the residential colleges where roommates and others were infected, then into the freshman dining hall where staff were infected, University Health Services where again staff were infected. We were seeing these events. The public health team was tracking them all, into commencement where individuals at commencement were infected and staff were infected, and later took the outbreak back to a local East Boston community. And we were able to actually see this through the public health data, but we sequenced the genomes of this and we could trace these things. That original outbreak in East Boston wasn't thought to have anything to do with Harvard. Our genome sequencing could show it was and could start working together. And the thing is, if you think about it, in an outbreak, university campuses are really affected because people are always sitting in lecture halls like on top of each other like this and dining together and all sorts of, they're sort of in an enclosed environment where infections can spread. And Boston is a great laboratory for that. It's got 60 residential colleges, 110 institutions of higher learning. And we saw through sequencing, the outbreak spreading between campuses and throughout and seeing relationships between them. But it also tells us that university campuses are also great places of thinking about how to respond. So the kinds of things that we can develop are mobile apps where we can use these paper-based diagnostics, let individuals start sharing their symptoms in real time, telling them, oh, by the way, 5 other people that were at that Broad 15 event that you have also come down sick. And one of them was just diagnosed with coronavirus. Maybe you should get tested. You could get that information in real time. Hopefully that's not the scenario that happens. I don't hear any coughing. It's great. But ultimately, those are the kinds of things you can have. There's a mumps outbreak that was like a paper about a mumps outbreak that was traced to a party. These are the kinds of events, but if you had that information in real time and you could actually use genomics to amplify that, it's really around genomics and information technology working together. And then, so hopefully we can get people to start speaking in real time, come to attention, create a system that encourages that supports them. We really want to empower every actor. We want to importantly empower frontline actors, and the Broad Institute has worked to train individuals from all around the world, but in lots of different programs. This is just one that we work with, and we worked with our collaborators in those West African countries, teaching them molecular biology, fundamentals of diagnostics, but then also to advanced sequencing and bioinformatics, extraordinary things through lots of different programs. But also the community. Ultimately, we don't actually have to wait for an outbreak to start to think about how to respond. And there's a really great way to teach how an outbreak would respond in middle schools, and that we've been working with in the Sarasota Military Prep Academy, which is Bluetooth on your phone. Essentially, you can spread a virus via Bluetooth on your phone and mimic every component of a real virus, the genome, changing in real time, the fatality rates and beyond. And so this is a program we've been running that's now been run in many schools in China and throughout the country, but really getting kids to be able to act and react and think about it, and we see all sorts of things. And fundamentally, the thing I've learned is these kids are amazing, and they really take on these roles, and they start tracking these outbreaks as they go. And the final thing we want to do is overcome, right? So we want to learn how to work together to position and to kind of communicate and recognize that we can kind of beat the clock, but ultimately we're waiting for some way of coming through. And genomics is important to that too, because ultimately antibodies that we use, the vaccines we use, are based on proteins that are based on the genome sequence, and we use that information to be able to create the best therapies and vaccines. But also, remember, CRISPR was designed and identified originally in nature as the bacterial immune system to viruses. And it is a way of taking a particular element that comes through, detecting it, and destroying it. And so there are ways of using that towards therapies. And so this is one of many ways that we are trying to advance that and that the community is advancing. And genomics can give you all sorts of new tools of thinking about new, completely novel approaches to doing therapies, looking through nature. And there's a vast amount of nature that we can probe to find new ways to overcome these diseases. So again, simply we're trying to detect, connect, engage, empower, and fundamentally what we want to do is work together to overcome. Because as Deb described, this sort of cases around focusing on the patient, and ultimately it's the same thing. That what we want to do is to be able to say if a child falls ill in a remote part of the world, anywhere, that we can actually create the tools to diagnose and treat them. And fundamentally, the most important thing is to save that child, but in doing so, we may be able to save a region and perhaps the planet from a catastrophic outbreak. Thank you.
Thank you very much, Pardis and Deb. And we are now at the Q&A portion of the evening. If you have any questions for our esteemed speakers, please raise your hand.
I'm creeping with you.
That's okay. Nope. I'll get out of your way. Or, like I said, if you're watching online, please start typing your questions into YouTube chat, or you can text your questions to the number that will hopefully be coming up on the screen shortly. Here we go.
Thank you for the excellent talks. How do you see the coronavirus epidemic playing out?
How do we see the coronavirus epidemic playing out? We're all tracing that ourselves and trying to understand, what is happening in real time. Fundamentally, as Deb described it, what's amazing about it is it was identified within days. There are diagnostics within days. They're moving. It's still not fast enough. We're just getting the diagnostics to do testing in the state of Massachusetts. There's been some questions about, there's been some little tricks in getting that. So up till now, only the CDC's been testing. That's not fast enough. So there's a lot we need to do to be able to test, isolate, and stop it. When you think about the virus itself, it's an RNA virus. It's related to SARS, and so there's some concern about that. It's also related to a lot of coronaviruses that about a quarter of infections every year that are detected are coronaviruses. So it could end up being something very typical. It could end up changing every time you see something new. You have to be concerned. And so fundamentally, I think what we still need to do is before is just to move as quickly as possible to be able to identify and isolate, and stop infections from going. But but I do feel as if the community. Ultimately, I don't even know if this virus is the big threat, but I do like the way that it's galvanizing the community to say, oh, wait, we need better diagnostics. We need them on the ground. We need to be able to think about this. And so if it helps us move the ball, I'm happy about it. And, always remain vigilant because you never know what will happen, but working towards it.
Thank you.
Yeah.
A couple of questions up over this way.
Thank you for the great talk. And Dr. Deborah mentioned that there would be the need for the larger international collaborations in order to fight the emerging antibiotic resistance. And do you think what would be the future role and the supragovernmental organizations like WHO should take?
What is the question? Can you?
It was basically like, how can we collaborate for antibiotic resistance, or what are the roles that was it governments can take, or?
Especially some kind of supragovernmental organizations like
Super governmental organizations.
Yeah. No, so as you know, as we've suggested, right, antibiotic resistance is a global problem. And so does it require collaborations between governments? Absolutely. You can see that how one country uses antibiotics and can generate resistance doesn't get isolated in that country because of travel and the globalization of our world. So it's interesting because there have only been a small handful of health issues that have been discussed in the UN, and antibiotic resistance is one of them because there is this recognition that you require actually incredible global collaboration in order to tackle it. So not only, I would go farther than saying, will that help? I think it's going to be mandatory for us to get it under control.
I saw another hand up back that way.
Thank you for your talk. I saw a video on PBS NewsHour a couple of nights ago about the Ebola virus outbreak in Congo currently ongoing. And they were mostly talking about the threat of sort of an anti-science, anti-medical feeling that was going on, medical people being attacked, and a lot of stuff going on around the internet. And I'm wondering if you saw that in West Africa when you were there, and is that a growing threat, or is that something how do we counter that sort of a thing that's going on? Thanks.
I mean, once again, I often want to say we have an anti-vaxxer issue in America. There's lots of reasons why people are suspicious of medical practice. And I think that the things that you can do, the best way you can do it is to just really work with those communities. And definitely you see some of this that happen in West Africa, but it's like when you quarantine people and you drag them away from their families and you don't tell them what's going on, I mean, everything, and then the people die anyway, there's of course reasons why people will be suspicious. And so I remember I wrote a briefing to somebody that was essentially like, we need to change the way we do this altogether, where we have to engage. I think at one of the times I said, Disney needs to do this, really. It needs to be something where people feel like when they're, and also in the United States, right, people were escaping quarantine because they felt like it was draconian, and it truly was. We trapped the Duncan family in their home, with all the soiled sheets. And so fundamentally, outbreaks are a time where it's a crucible. It's a deadly virus is spreading within a population. Everyone gets suspicious and misbehaves. And that will lead to all of this mistrust. And that's on top of political instability that you see in DRC already. It gets worse. And if you're going to try to respond to this, you have to do it with care. You have to understand it's the virus is weaponizing people in a way, but it's the person is human. And if we figure out how to help each individual get better, the community will solve itself. And so I think it's a more fundamental problem that happens in every country in some different way that is exacerbated when there's a political turmoil. And it particularly happens to be bad in the DRC, but it's a fundamental problem. And I imagine if an outbreak spreads to the United States, you'll see people punching each other in grocery stores for cans. And we have to not do those things. We have to figure out how to work together.
All right, I think we have time for one more question, and I saw a hand up over here.
Hello, great talk. I have actually two questions. One is the current coronavirus outbreak. So according to the Chinese scientists, there are a lot of false negative if we use the current testing kit. I believe is the RT-qPCR. So is there any way to circumvent this diagnostic things? Another question is this current method takes a much longer time, and you mentioned this Sherlock is much faster. So I wonder if there's any company or Broad is developing the tool for diagnostic.
I'm actually really glad you said that because this is a more popular talk. Our general conflict of interest disclosure was not there, and I didn't say this from the outset, but Deb and I are both co-founders of a company. So you can take everything we said with a grain of salt and say all of it's garbage. I have no problem with that. I have no credibility because we do have an investment in a company, but that brings that Sherlock technology to bear. So I'll just start by saying, I apologize for not saying that from the outset, but that's very important. And we're doing it fundamentally because we think it's important. And we realize that a lot of times in order to move things forward, it's through companies. That's the way that our research tools are not going to get to where they need to go. But yeah, there are many different diagnostic tests that are coming out that are, PCR is fantastic, and it's great because you can develop diagnostics quickly, within days, and get them out into the world. But they are big machines, and they take a while. And yes, you can, and they often are not multiplexed, and so they can't capture a lot of different diverse strains. CRISPR is one of many different approaches that allows you to get to things that could be paper-based, that could happen faster. And so it's a new and burgeoning field. There's synthetic biology tools as well that are coming out of MIT and elsewhere. But are in everywhere. Like everywhere people are developing these kinds of tools, in Berkeley and elsewhere. And so there's a real move to that of transforming those types of diagnostics. And false positives and false negatives are often an issue, and that's another component piece that they're trying to work through. Yeah.
Well, thank you for that question. Thank you, Pardis. Thank you, Deb. And thank you to all of you for joining us tonight.